Hello everyone. Welcome to today's live class. Okay. In today's class, we're going to discuss about the main important topic that is the cell membrane. Cell membrane is one such an important uh, part of the cell, and this actually sets a boundary. And this plasma membrane is a security gate of the cell. Means what? It protects the cell carefully, and it controls the movement of molecule across what the boundary. Okay, plasma membrane. In today's class, we're going to discuss about the important headings like the structure of plasma membrane and the chemical composition of the plasma membrane, and the most important, the fluid mosaic model is what mainly given by the two scientists Nicholson and the Singer and Nicholson in 1972. And finally, we're going to discuss the functions of the plasma membrane. Now, firstly, uh, let me draw the diagram of the plasma membrane. Okay, let me take another slide. Yes. Now we're going to draw what? The diagram of the plasma membrane. That is structure of plasma membrane. Before going to understand the structure, you must know that the plasma membrane also called as the cell membrane, and it's also called as cytoplasmic membrane. Okay, all the three names are for one, that is the plasma membrane. Now, let us discuss the structure. Before going to that, let me construct the diagram. First, we're going to observe what? The lipid bilayer. Lipid bilayer. Along with me, you also go to draw the diagrams. This is lipid bilayer. Yes. Now, let me take another color. Yes. I'm going to construct the important protein structure, that is integrated protein. This is integrated ಅಥವಾ integral proteins. Integral protein. And the another one is the first one, what actually we constructed. This is a lipid bilayer, but in the form of phospholipid bilayer. And each phospholipid bilayer having two important structures, that is head structure and the tail structure. We can observe here lipid. In the lipid bilayer, each layer consists of lipid molecule, and lipid molecule consists of what? The two parts. One is a head part, another one is a tail part. Head part and the tail part. Yes. Uh, and this is what we call head, and this is tail, and this is okay. Let me erase something here. I don't want to put you in confusion. Yes, this one is called integral protein. And another structure we're going to observe, And this structure is also what protein, but this is not completely embedded in the lipid bilayer. They are floating here and there and outside the lipid bilayer, hence they are called peripheral protein. Hence they are called peripheral protein. The another important structure along with this, the some carbohydrates, they also we're going to observe in this. The carbohydrates is actually two form. They exist main in the what the complex form or in the conjugated form. The first one is this is glycolipid and the another one is the same but it is somewhat different. It's a glycoprotein. Glycoprotein. And the glycolipid may be or the glycolipid or the glycoprotein may be straight chain like this or branched chain. This is a straight chain. This is a branched chain. And copy this diagram and this is the structure of what? The plasma membrane. Okay. Okay. Now let me discuss or explain this diagram. This is a diagram of the plasma membrane. Okay. Before going to that, the term plasma membrane is coined by Nageli, the scientist Nageli and Kramer in 1855. Okay, the term plasma membrane is given by or it is coined by the scientist Nageli and Kramer in 1855. Now, the let me discuss one by one that in structure we are going to first understand the chemical composition. The chemical composition, let me take the different color, yes. Chemical composition of plasma chemical composition of the plasma membrane. Okay, so hereafter I don't want to write the complete as like plasma membrane. I just mention as the PM, and don't get confused with the prime minister. This is a plasma membrane. The chemical composition of plasma membrane, mainly going to observe what the chemicals in the plasma membrane. You can uh, list by observing this diagram which chemicals are mainly we're going to observe in the plasma membrane. The first chemical is that is a lipid. That's a phospholipid means what? The lipid. Lipid is a one chemical. Another one is called the protein. Yes, protein also we're going to observe. And the third one, glyco. Here also glyco and here also glyco. Glyco refers to what? Carbohydrates. The carbohydrates. Okay. All these are what? They may be in the simpler form or they are in their conjugated form. But they are what we're going to observe. All this chemical together they constituted the plasma membrane. Okay, now firstly I going to explain the lipid molecule. Lipid molecule, you can take this molecule. Let me draw the diagram in another slide. The lipid composition. The first one is what? Lipid. The lipid molecule, when we take, yes, this is the structure of the what? This is the structure of, yes, the lipid, phospholipid. This is a structure of phospholipid molecule. Phospholipid molecule. The phospholipid molecule mainly consists of what? Here, as name indicating, this is a phosphate group. This is a phosphate group, and this is the glycerol part. Glycerol part. Glycerol, why we're going to observe is as a lipid, as it is a lipid molecule. Glycerol, we know that it is the backbone of the lipid. Hence, we're going to observe glycerol molecule, and here the fatty acids. This straight chain like chain indicating that it is a saturated fatty acid, and here this is a kink. Kink means what? Bend. This bending, and this is what? Unsaturated fatty acid. Okay. Now let me clear first this one. The plasma membrane contains about 20 to 80% that is made up of lipids. Did you get the point? That plasma membrane in majority of its chemical is what? The lipid. It constituted about 20 to 80%. The several lipids present in the plasma membrane. And the lipid molecule, and this is what the chemical structure I already drawn here. That's the each lipid molecule made up of chemicals, like one is the glycerol, phosphate group, and the unsaturated fatty acid, unsat- unsaturated fatty acid, third one, and the fourth one is the saturated fatty acids. But in the physical structure, we're going to consider this molecule are divided into two parts. The first portion is called, and let me draw here again. If I write overlapping, this is head structure and this is tail structure. Head is head of the lipid molecule is hydrophilic in nature and the tail is hydrophobic in nature. Now the question arises, the single lipid molecule, but it has two parts. One is a hydrophilic and hydrophobic parts. And that's why the lipid molecule is called amphipathic molecule. Amphipathic molecule or amphipathic nature. Amphipathic in nature. And now the question arises, why tail is hydrophobic and head is hydrophilic in nature? Head is the hydrophilic. Why? Because along with the glycerol, we can observe here, the answer is here. The glycerol is what the water insoluble portion, but the glycerol and the fatty acids together the lipid molecule, but the phosphate group which makes the head portion as hydrophilic in nature. Okay. Okay, now I am going to repeat the same again. The lipid from the diagram of the plasma membrane, this lipid molecule, when we enlarge, it appears like this. It appears like this. And this is the head portion, which is spherical in nature, and two long tail-like structure, they are called tails. And chemically, head is made up of two different chemicals. One portion is a phosphate, another one is a glycerol. And the two tails are also different in nature. One is a saturated fatty acid, another one is a unsaturated the fatty acid. Okay. So unsaturated fatty acid, you can observe here the kink means bending. Why it is bend means here there is no single bond, hence it appears to be straight line. But here there may be double bond or triple bond. So that's why here we're going to kink. Kink means what? The bend region. Okay. This is the, yes, lipid, the chemical of the plasma membrane. The next one is the proteins. Next one is the proteins. Here proteins in the plasma membrane. In the plasma membrane, they appear in two different forms. They appear in two different forms. One is that is integrated protein. Integrated protein. The another one is peripheral protein. Integrated protein means integrated protein, you can observe here, this is the integrated protein which is completely embedded in the lipid bilayer. It is completely embedded in lipid bilayer. Lipid bilayer. Okay. And some portion of this integrated protein they expose to the outer side, means to the periphery, or majority of this protein part that is completely embedded in the lipid bilayer. You can observe in the diagram. See here, this is an integrated protein. Its major portion is embedded in the lipid bilayer, and this integrated protein, what maybe it sometimes serves as a specialized protein channels. Protein channels. They acts as a protein channels for the movement of specific molecules. For the movement of specific molecules. Okay, so therefore such specific protein channels they are referred as a channel proteins, channel protein or transport protein, transport protein. The next one is a peripheral protein. The peripheral protein means, you can observe in the diagram here, this is a peripheral protein. This only the little portion or some portion of its protein is embedded in the lipid bilayer, or maybe that is completely outside of the lipid bilayer. They are situated outside the lipid bilayer. Means that peripheral region means what? Outside or the out towards the outside. Okay. Hence it is called peripheral protein. Hence it is called peripheral protein. Now this is a protein, the another chemical. The protein mainly they are of what? Yeah, exist in two form. One is the integrated protein and the peripheral protein. Integrated protein sometime based on their specific work they are referred as a transport proteins. Okay. Now another, let me move on to the another chemical component. The third one is that is carbohydrates. Is the carbohydrates. The carbohydrates also in the lipid, in the plasma membrane appears in two different forms. You can observe here the first one is glycoprotein, the second one is a glycolipid. Okay, appears in how many forms? Two different forms. One is glycolipid, second one is a glycoprotein. Okay. These are the two different forms of the carbohydrates. The glycolipids means they are covalently bonded to, sorry, lipids. Here is, yes, glycolipid means lipids, they are covalently bonded with the carbohydrates. Hydrates. Okay, they located exclusively on the extracellular leaflet of the what plasma membrane. Means you can observe in the diagram they are not embedded in the lipid bilayer. Okay, they are situated outside the plasma membrane. Okay, so that is what we referred as a extracellular presence. And these glycoproteins they involve in what? Cell recognition. Cell cell recognition. Cell recognition. What actually the meaning of cell recognition? The cell to cell recognition means the process where cells identify the neighboring cells. Each cell must identify what their sorry identify must identify the neighboring cell. So this is called cell to cell what identification means the glycolipids on the plasma membrane of particular one cell and on the other cell they are going to identify the neighboring cells by identifying this particular the glycolipids. Now come to the okay. Next is another one that's a glycoprotein. Glycoprotein, they are nothing but, let me change the color here. Yes. Glycoprotein, nothing but the proteins. They are covalently what binded with the carbohydrates. Okay, the combination of proteins, glyco refers to what? The carbohydrates. Here protein and the carbohydrates, how they join together by a covalent bond, and this conjugated structure is called glycoprotein. Glycoprotein primarily their function is to cellular, they acts as a cellular IDs means cell identification. They involved in what? Cell identification, cell identification, and they serves as a receptor for, they serves as a receptor for what? For chemical signaling. For chemical signaling. Okay, chemical signaling and adhesion molecules that bind cell together, and they involved in what? Cell to cell binding. Cell to cell binding. Okay, this is about the carbohydrates, and these are the three chemicals which are mainly we're going to observe in the plasma membrane. Can you repeat that? The first is called the lipids, and the second one is the proteins, and the third one is the carbohydrates. Yes, that is the phospholipids are mainly the here I just forgot that to mention the function of this plasma membranes lipid, that's a phospholipid molecule. These molecules mainly acts as a selective permeable barrier. They acts as a selective permeable barrier, means they restrict the movement of molecule across the this membrane. Okay, and these provide structural integrity. They provide structural integrity, and they maintain these phospholipids. The phospholipids they maintain what? The membrane fluidity, and they also serves as a are involved in cell signaling, and these phospholipid they involved in the cell's dynamic activities like exocytosis and endocytosis. Okay, now we're going to observe the function of integral protein. Yes, here we already mentioned that the integral protein they are what acts as a transport protein and acts as a channel protein and protein pumps, and they form what? Protein forms the protein pump to transport the electrolytes or other particular molecules, and they helps in what? Yes, enzymatic activity. They involved in enzymatic activity, and they provide structural support, and these they provide what? Structural support. In the same way, the peripheral protein peripheral protein they perform their particular functions. Mainly their core function in the plasma membrane are they serve as a bridge points. They serve. Okay, let me take another slide. That is the functions of peripheral protein. The peripheral protein they serve as bridge bridge points. Okay, and why they we are call it as a bridge points means they are connecting the plasma membrane to the the cytoskeleton cytoskeleton, such as actin filament. We can take example the actin filament. And second one is they also involved in what? Cell signaling. Cell signaling means they acts as what? Signal transductors. Signal transducers. And they peripheral proteins are they are sometimes they act as enzymes. Okay, they maintain the structure of the plasma membrane. And the fifth point is they involved in electron transportation. Electron transportation. Okay, so all these are the functions of particular what. For example, we discussed the functions of lipid molecules also, that is phospholipid molecule, and the integral protein and the peripheral protein and the carbohydrates. In the carbohydrates, glycolipids and the glycoproteins. All things we have here discussed. Now we're going to discuss about the fluid mosaic model. The fluid mosaic model, the important another heading, that is a fluid mosaic model. The fluid mosaic model first actually explained by two scientists. One is the Singer and Nicholson in 1972. According to them, according to them, uh, mainly to explain the plasma membrane, you they explained like the core postulates of core postulates of the fluid mosaic model. The first one is, according to them, the Singer and Nicholson, the plasma membrane is made up of lipid bilayer, and this lipid bilayer they provide fluidity to the membrane. Okay, the first one is the they explained that plasma membrane is made up of lipid bilayer. We already discussed that. Lipid bilayer means this structure you already observe, and this because of this arrangement, this provides the fluidity to the plasma membrane by according to these two scientists. And mosaic arrangement, they explain what? mosaic arrangement. What actually is the mosaic arrangement? Means the membrane is a mosaic of various components. Means the plasma membrane is mosaic of various components. Various components. Means this the plasma membrane is what chemically composed of. We can understand like this: mosaic of various components. Mosaic means mixture of. We can understand that is a mixture of the chemicals. Like, can you list out? We discussed this earlierly. That is the first one. The plasma membrane is made up of lipid, that is in the form of phospholipid. Second one is a protein, and the third one is a lipid, protein, and the yes, carbohydrates. Carbohydrates. Okay. Okay. Now the integral and peripheral proteins. The next point is the integral and peripheral proteins. Means the protein is in two forms. One is the integral protein form, and another one is a peripheral protein form. Okay. So they explain in the fluid mosaic model the role of cholesterol. Role of cholesterol. Mainly the cholesterol, particularly in animal cell. Animal cell, in animal cell, cholesterol molecules are tucked between the phospholipids. How can we understand? Means if you consider this is a lipid bilayer, yes, and this is a tail structure, this is a head structure. Now let me take another color. Yes, according to the Singer and Nicholson, here what the cholesterol molecule that is stuck between the phospholipid molecules. Okay, and it acts as a fluidity buffer. It acts as fluidity buffer, preventing the membrane from becoming too fluid at high temperature or too rigid at low temperatures. And finally, Nicholson and Singer, they explain that the plasma membrane is selective permeable in nature. They explain the plasma membrane is in selective permeable nature. Due to its fluid and chemical nature, the membrane acts as a semi-permeable barrier that controlling which substance enter and which substance exit the cell based on size and polarity. Okay, so all this about the postulates that is given by the Singer and Nicholson. Now let me move on. Another important point, that is the function of the plasma membrane. Okay, in next class we will discuss about the fluidity and function of plasma membrane.